Hi students, today the class is about factors promoting neuronal growth. It is divided majorly into two groups. They are neurotrophins and other factors which are affecting the neuronal growth. The neurotrophins includes NGF, that is nerve growth factor, PDGF, that is brain derived growth factor, NT3, that is neurotrophin 3, and NT45, that is neurotrophin 4/5. And other growth promoting factors are CNTF, that is ciliary neurotrophic factor, LIF, that is leukemia inhibitory factor, IGF-1, insulin like growth factor 1, TGF, transforming growth factor, FGF, fibroblast growth factor, and PDGF, that is platelet derived growth factor. So we will discuss about all these things but not in detail. Okay, we will discuss the main points only. About neurotrophins, we will go to some extent. The discussion will go to some extent, but regarding the other growth promoting factors, we will only discuss the important ones, important points. First, coming to the neurotrophins. What are neurotrophins? These are the family of proteins that will induce the survival, development and function of the neurons. They belong to class of growth factors, secreted proteins. These are the secreted proteins that can signal particular cell to survive, differentiate and grow. Growth factors such as neurotrophins that promote the survival of the neurons are also known as neurotrophic factors. The neurotrophic factors are secreted by target tissue and act by preventing the associated neuron from initiating programmed cell death, and it will allow the neurons to survive. So these are secreted by target tissues and it protects the neuron. Okay, and it prevents the apoptosis, that is it prevents the programmed cell death in the particular neuron. Neurotrophins also induce differentiation of the progenitor cells. So they will form the new neurons. Although the vast majority of the neurons in the mammalian brain are formed prenatally, that is before birth, parts of the adult brain, for example the hippocampus, it will retain the ability to grow new neurons from neural stem cells. This process is known as neurogenesis. These neurotrophins are chemicals that will stimulate and control the neurogenesis. The term neurotrophin may be used as a synonym for neurotrophic factor. So, neurotrophic factor or neurotrophins are same. See this transport of neurotrophins? They are transported into the neuron retrogradely. It is called retrograde transport. So, they are released by target cells and they are going from axon terminal via the axon to the cell body. It is a retrograde transport. Okay. So if there is no neuro target derived neurotrophic factor, so this entire neuron is undergoing apoptosis. So in the case of neurotrophic factor supply, this is a healthy neuron. If the neurotrophic factor is deprived, then it is a sick neuron. In neurotrophins, the first one is the NGF or nerve growth factor. It was first isolated by the Nobel laureates Rita Levi-Montalcini and Stanley Cohen in 1956. Okay, so Rita Levi-Montalcini co-discovered the nerve growth factor with the Cohen in 1956. Okay. And they got Nobel Prize in 1986 in physiology or medicine. It is a prototypical growth factor. That is, it is the first to be identified and studied in detail. Coming to the structure of the NGF. NGF is initially a 7s 130 kilodalton complex of 3 proteins. So it is composed of 3 proteins, that is alpha, beta and gamma NGF in 2:1:2 ratio when it is expressed. This form is also known as pro-NGF or NGF precursor. So this alpha, beta and gamma together known as pro-NGF or NGF precursor. Coming to the gamma subunit, the gamma subunit of this complex has a activity like a serine protease. So it has a activity of serine protease activity, and it cleaves the N terminal which is present in the beta subunit. So our pro NGF will be activated to functional NGF. So the term actually nerve growth factor refers to the functional NGF, which is 2.5s 26 kilodalton beta subunit of the protein. Actually, the middle one, beta subunit of the protein, is the active ingredient. Okay, it is the only component of the 7s NGF complex that is biologically active. That is, this beta component will act as a signaling molecule. Now coming to the functions of NGF. This NGF is involved primarily in the growth as well as the maintenance, proliferation and survival of the neurons. And it is critical for survival and maintenance of sympathetic and sensory neurons, as these neurons will undergo apoptosis in its absence. So for growth and survival of sympathetic neurons and sensory neurons, they will need NGF. However, recent studies suggest that NGF is also involved in pathways, those regulating the life cycle of the neurons. So the main three important points are: it is involved in the neuronal proliferation and proliferation of the pancreatic beta cells and regulation of the immune system. So this NGF is important not only for neuronal proliferation but also for a proliferation of the pancreatic beta cells and a regulation of the immune system. So we have discussed the NGF who has done research on it and who has identified this NGF factor, two scientists, and they have got a Nobel Prize also. And we have discussed the structure. Okay, structure also we have discussed, and we are discussing the functions, that is neuronal proliferation, that is this actual NGF will act through TRKA receptor, that is tropomyosin receptor kinase A or tyrosine receptor kinase A. Okay, with the help of this it will stimulate the survival and proliferation of the target neuron. Okay, so in the function of neuronal proliferation, because of the binding of the NGF with the TRKA receptor, stimulate the proliferation and survival of the target neuron. Okay, mainly through the expression of the genes. What is that gene? Especially BCL2 gene. So with the help of this signaling, what happens in the nucleus, BCL2 receptor, sorry, BCL2 gene is expressed, and this leads to the proliferation and survival of the neuron. Okay, so it has high affinity of binding for TRKA receptor, and it is also having affinity for P75 NTR receptor. We will discuss about these receptor later on. Okay, so mainly neurotrophins will act through TRKA receptors. We have subtypes and P75 NTR receptors. So if they act on TRKA receptor, they will stimulate the proliferation of the neuron. If they act on P75 NTR receptor, they can lead to programmed cell death or survival. Okay, that is the action of. So we will come later on what is the detailed action of these receptors and with these neurotrophins with the receptors. Then coming to the proliferation of pancreatic beta cells. So our pancreatic beta cells will express both TRKA receptors and P75 NTR receptors. So on both these receptors, NGF will be acting. Okay. So if you withdraw the NGF, what happens? It induces apoptosis in the pancreatic beta cells. So it signifies the critical role which is played by the NGF in the maintenance and survival of the pancreatic beta cells. Now coming to the regulation of the immune system. This NGF will play a critical role in regulation of both innate immunity and acquired immunity. So in the process of inflammation, this NGF is released in high concentration by mast cells. Okay, mast cells, and they will induce the axonal growth. Okay, axonal growth in the nearby nociceptive neurons. Nociceptive means which are taking pain sensations. This leads to increased pain perception in areas under inflammation. So in inflammation, why we are perceiving more pain? That is because of more amount of NGF is released by mast cells at the site of inflammation, and this NGF is leading to proliferation of the nociceptive neurons at that point. So that is about inflammation, and so that is about innate immunity. And in acquired immunity, this NGF is produced by thymus cells as well as CD4 plus T cells. Okay, so in innate immunity, it is produced by mast cells. In acquired immunity, NGF is produced by thymus cells and CD4 plus T cells, and it will induce a cascade of maturation of T cells under infection. So it is helpful for T cell maturation in acquired immunity. And this NGF, so recent studies which are made on this NGF, they revealed that this NGF is abundantly present in the seminal plasma. Okay, and it induces ovulation in some mammals. Example, induced ovulators like llamas. Okay, so it was previously called as OIF, ovulation inducing factor in the semen. Okay, actually it is a beta NGF. So that beta form is actual active form. See this signaling mechanism by NGF. So this NGF will act on both TRKA receptors and P75 NTR receptors. It has high affinity for TRKA receptor and low affinity for P75 NTR receptors. Okay, and these both are associated with some neurodegenerative disorders. So what happens when NGF is binding to TRKA receptor? So after binding of NGF with TRKA receptors, homodimerization. This is one receptor. Then this is other TRKA receptor. Homodimerization of the receptor happens. Then it causes autophosphorylation of tyrosine kinase segment. That is, it is attaching phosphate into the same into the receptor. That is autophosphorylation. Okay, it is phosphorylating by itself to the where it is phosphorylating to the tyrosine kinase segment. Then what happens? The TRKA receptor actually has 5 extracellular domains. You see 1, 2, 3, 4, 5. 5 extracellular domains, and the fifth domain is sufficient in binding TRF, binding NGF. So fifth domain is itself actually is sufficient. So once bound, what happens? This entire complex undergoes endocytosis. Okay, endocytosis means it's coming into the cell. Endocytosis and activates the transcriptional program, which is following two major pathways or three major pathways. So first pathway is a RAS MAPK pathway, and the second pathway is the PI3K AKT pathway. And sometimes uh, it also follows PLCY pathway. Okay, so this so up to what thing we have discussed? Binding, then homodimerization, then autophosphorylation, then internalization, that is endocytosis happened, and this will stimulate all these mechanisms. So these are called adapter proteins. Adapter proteins. So this is one major pathway is AKT pathway. Okay, one major pathway is the activation of the AKT. AKT is a kinase. It is also a kinase, serine threonine kinase. Okay. Now this pathway, what happens? Activation of the AKT. After the activation of AKT, what happens? Recruitment of a second protein occurs, that is GRB2. Okay, along with the GAB1. So this AKT and GRB2 are they are adapter proteins. These will along with GAB1 protein, that is a docking protein. It will be attached to that, and it will cause survival of the neuron. If it is excess, it will lead to all these things. And PI3K, that is phosphatidyl inositol 3 kinase, is activated, resulting in AKT kinase activation. Okay. If you block this one, if you block phosphatidyl inositol 3 kinase AKT pathway, okay, what happens? Death of the sympathetic neurons occurs. If NGF is also present, but you are blocking this pathway, then what happens? Sympathetic neurons will die. Now coming to the second pathway, RAS MAPK pathway. MAPK means mitogen activated protein kinase pathway. In this pathway, what happens? There is activation of the RAS. It is a G protein. Okay, it is a G protein. Then what happens? RAS is activated, then MAPK is activated, and this facilitate the transcription. Okay, and proliferation of the neuron. Some in some cells, PLC-Y pathway will be present. It leads to synaptic plasticity. Then it will also act on the P75 NTR receptor. Here also actually three major pathways are there. If NFKB pathway is activated, then it leads to survival of the cell. If JNK pathway is activated, it leads to apoptosis of the cell. And this NGF is also present in basal forebrain and striatum of cholinergic neurons. So it is mainly present in the cholinergic neurons which are present in the basal forebrain and striatum. Okay, and it is related to Alzheimer's disease here. How it is related? If there is no conversion of pro NGF to NGF, or this mature NGF is undergoing degradation rapidly, then it leads to the Alzheimer's disease. Okay, so here pro NGF to mature NGF plasmin is helping. Okay, and because of MMPs, it is undergoing rapidly degradation if they are present more. So there is occurrence of Alzheimer's disease. We are not going into detail about Alzheimer's disease pathology. Antiserum against NGF, if injected in a newborn animal, it causes immunosympathectomy. Okay, so if you inject antiserum against the NGF, where into the newborn animal, it causes immunosympathectomy. That is, complete destruction of the sympathetic ganglia occurs. Okay. So we have discussed all points about the NGF. Now coming to the second one, that is BDNF, brain derived neurotrophic factor, or it is also called as abrineurin. BDNF is a protein in humans and it is encoded by BDNF gene. It is also a member of neurotrophin family and it is related to NGF and NT3, NT4 and NV5. It is found, these neurotrophic factors are actually found in the brain and periphery. This BDNF was first isolated from a pig brain in 1982 by V.S. Alain Bardet and Hans Theunen. This BDF acts through TRKB tyrosine kinase receptor. Coming to the functions of the BDNF, this acts on certain neurons of the CNS and PNS which are expressing TRKB receptor, and it helps to support the survival of the existing neurons and encourages the growth and differentiation of the new neurons and the synapses. So it helps in the formation of synapses and growth of the new neurons and differentiation of the new neurons. It is mainly helpful for survival of the existing neurons. In the brain, it is active in the hippocampus, cortex, basal forebrain. So these are the areas which are vital for learning, memory and higher thinking. And it is also expressed in retina, kidneys, prostate, motor neurons and skeletal muscle. It is a growth promoting role. It is playing a growth promoting role for mainly peripheral sensory nerves. Peripheral sensory nerves. So if you remove the BDNF, then peripheral sensory nerves have not developed. and there will be severe degenerative changes. Certain types of physical exercise, it will increase the BDNF approximately by threefold. Okay, this is a phenomenon which is partly responsible for exercise-induced neurogenesis and improvements in cognitive function. So, how exercise is improving the neurogenesis and cognitive function? Because of the BDNF. That is, physical exercise causes skeletal muscle contraction. This skeletal muscle actually releasing irisin, and it is helpful for formation of BDNF. And this BDNF is leading to increased cerebral blood flow, increased brain volume, that is hippocampal volume, thereby increasing cognitive function and increasing synaptic plasticity and causing neurogenesis. About irisin, you see it, physical exercise, muscle, it is releasing irisin. This irisin is a kind of muscle cytokine. It is secreted from muscle, skeletal muscle. It is secreted by skeletal muscle under severe exercise. And exercise frequency, mode of exercise, and intensity of exercise will affect the release of irisin. So, severe exercise will release the irisin. And acute exercise can also increase the concentration of the irisin immediately. Where chronic exercise will affect the kinetics, that is metabolic kinetics and secretion efficiency of the irisin by skeletal muscle. So, acute and severe exercise will release more irisin. So, it will act through STAT3 signaling on the hippocampus. It leads to hippocampal proliferation. Hippocampal proliferation is acquired from irisin-induced STAT3 signaling modulation. And what happens? Physical exercise increases the BDNF synthesis in the brain, and irisin is also increasing BDNF synthesis. These are leading to neuroplasticity and decreased Alzheimer's disease risk. So exercise releasing FNDECS, that is, these are cell membrane proteins, and upon proteolysis they will give irisin. And this irisin is acting as a bridge between exercise and neurological diseases. And this is acting on the muscle brain axis. So it is a component of the muscle brain axis. Not only this irisin is important for cognitive functions, but is also causing many other functions. What are they? It will decrease obesity, and it will decrease insulin resistance, and it will decrease food intake, increases weight gain, and increases lipolysis, and it decreases gluconeogenesis and decreases blood glucose levels, increases glycolysis, and increases glycogenesis. Coming to the third one, that is neurotrophin 3. It is for the development of proprioceptive neurons and some subsets of mechanoreceptor sensory neurons. Okay. So NGF is for sympathetic and some sensory neurons. BDNF is responsible for peripheral sensory neurons, and neurotrophin 3 is a neurotrophin 3 is essential for proprioceptive neurons and some subsets of mechanoreceptor sensory neurons. It supports the survival and differentiation of the existing neurons, and it encourages the growth and differentiation of new neurons, and it causes new synapses to be formed. It is having ability to activate two kinds of receptors, that is, it can act on TRK B receptor and it can act on TRK C receptors. Next, coming to the last neurotrophin, neurotrophin 4-5. It is a neurotrophic factor that signals predominantly through TRK-B receptor tyrosine kinase. So it will act through TRK-B receptor. This was discovered from Xenopus and viper in 1991 by Finn Halbuk et al. It has neuroprotective effects, and it is for the development of motor neurons and also for development of retinal ganglion cells. and also helpful for survival differentiation of embryonic striatal neurons. Now coming to the receptor level. Now we have completed 4 neurotrophins, that is NGF, BDNF, neurotrophin 3, neurotrophin 4 by 5. Then we came to TRK receptors. What is TRK? Tyrosine receptor kinase or tropomyosin receptor kinase receptors. These are having three in this family. Three are included. That is TRK A, B, C. Okay, neurotrophins receptors and their signaling. So this is a homodimerization. Okay, P means phosphate. It can autophosphorylate, and these will, so the NGF will act on TRKA, BDNF NT4 will act on TRKB, NT3 will act on TRKB and C. These can act by these three pathways. That is PI3K pathway, phosphatidylinositol 3 kinase, and leads to the activation of some adapter proteins. Then it causes activation of inositol phosphate 3 kinase, and it leads to the activation of AKT kinase, which is leading to cell survival. And RAS pathway, it is causing activation of RAS, and it is causing activation of MAPK, mitogen activated protein kinase. It leads to neurite outgrowth and neuronal differentiation. And through phospholipase C pathway also, it is releasing IP3 and DAG and causing increased PKC and calcium in the cell, causing neurite outgrowth and neuronal differentiation. See the structure of the TRK receptor. This is one TRK receptor, A, B and C. So all TRK receptors are type 1 receptor tyrosine kinases. They have an extracellular ligand binding domain. So this is the extracellular region, this is the intracellular region, this is the cell membrane. So extracellularly we have ligand binding domain, that is NGF, all these neurotrophic binding domain, that is a ligand for that. And next it is having a single transmembrane domain, and it is having a cytoplasmic domain which is having a tyrosine kinase catalytic activity. So this is the structure of the TRK receptor. Coming to the P75 NTR receptor. The neurotrophins have long been known to promote the survival and differentiation of the vertebrate neurons, we know. But these growth factors can also induce cell death by acting on P75 receptor. It is a member of TNF, that is tumor necrosis factor receptor superfamily. This P75 receptor is mainly expressed during early neuronal development. In adult, it is re-expressed in various pathological conditions, including epilepsy, axotomy, and neurodegenerative conditions. Okay, potentially toxic peptides like amyloid B peptide that accumulates in Alzheimer's disease. This is the ligand for P75 receptor. So, just a point about AD, that is Alzheimer's disease. In this disease, amyloid B is accumulated. It is a ligand for P75 receptor. In recent work shown that P75 is functioning in the regulation of both synaptic transmission and axonal elongation. It works with the no-go receptor. It is a binding protein for axonal growth inhibitor and appears to be transducing subunit of this receptor complex. So, our P75 receptor, it is working with the other receptor, that is a no-go receptor. It is responsible for inhibition of the axonal growth, and appears to, this no-go receptor appears to be a subunit of the P75 receptor. See this P75 receptor and its co-receptors. This is a P75 receptor. N-terminal, C-terminal. C-terminal is having chopper domain, death domain. Okay, gamma secretase will cut here. It will cut the chopper domain, death domain from the N-terminal end. Okay, this is N-terminal which is having cysteine rich domain 1, 2, 3, 4. and it will work with the other receptors like our TRK receptors, sortilin receptor. This is a no-go receptor. Okay, it will work with other receptors. So what is the schematic view of this receptor? It is having an extracellular N-terminal part, and this extracellular part is having four cysteine rich domains, and then it is having a single transmembrane domain, and it has an intracellular C-terminal domain. This intracellular C-terminal domain is having chopper domain and the death domain. The extracellular part is cleavable by TACE or ADAM17 enzyme, while the intracellular part is cleaved by gamma secretase enzyme. P75 receptor has several co-receptors we have already seen. What are those? Sortilin, TRK, ABC and no-go receptors. Signaling pathways by neurotrophins. See this? P75 will act through three mechanisms and TRK receptor will act through three mechanisms. What are those mechanisms? So this picture actually depicting the interactions of the each neurotrophin with the TRK and P75 receptor, and it is showing major intracellular signaling pathways associated with these receptors. Coming to our P75 receptor, it regulates three major signaling pathways. What are they? First one is the NFKB activation. This NFKB activation results in the transcription of the multiple genes, and it causes survival of the neuron. It promotes the survival of the neurons. Next pathway, activation of the Jun kinase. So this Jun kinase pathway, if it is activated, it controls the activation of several genes, but it will cause apoptosis. So it will promote neuronal apoptosis. And third pathway is activation of the Rho. If this pathway is activated, it will control the growth of the neuron. That is, it will inhibit the growth of the neuron. So through NFKB activation, it is causing promotion of the neuronal growth. Where Jun kinase and activation of the Rho mechanism is causing controlling of the neuronal growth. The pro-apoptosis actions of P75 receptor appears to require presence of sortilin, which functions as a co-receptor. So these two will require presence of sortilin receptor also. Sortilin we have seen in the previous figure, not in this figure. Now coming to the TRK receptor. Each TRK receptor controls three major signaling pathways. What are those? MAP kinase pathway. So activation of the RAS will cause activation of the MAP kinase. It will cause neuronal differentiation and neurite outgrowth. Next pathway is the PI3K pathway. Okay, this activation of the PI3 pathway through RAS or GAP1. So all these are adapter proteins. So PI3 kinase is activated because of these molecules activation. This will promote the survival and growth of the neurons and other cells. Now coming to the third pathway, that is activation of the PLC gamma 1 results in activation of the calcium and protein kinase C. It will promote the synaptic plasticity. Okay. Then each of these signaling pathways also regulates gene transcription. Many additional adapters for P75 and TRK receptors are identified, which are not shown in this figure. So we have shown the main pathways only. All these are RAS and these are adapter proteins, and this RAS is a G protein and this GAP1 is a adapter protein. So not only this is involved, but many adapter proteins are involved in all these pathways. Now we came to other growth promoting factors. So what are the other growth promoting factors? First one is the ciliary neurotrophic factor, that is CNTF. It is important for spinal cord neuron development. Okay, neurons which are present in the spinal cord, they require CNTF. And not only that, it is also involved in the bone activity. That is, it reduces the osteoblast activity in the bone. And It is used for treatment of motor neuron disease conditions. It could and not only that, it could reduce the food intake without causing hunger or stress, making it a candidate for weight control in leptin resistant subjects. Okay, so in the case of leptin resistant subjects, if they are obese, then we can try CNTF. So recombinant of human CNTF is available. It is called axokine. It is useful for obesity reduction. So the P53 regulates the maternal reproduction at the implantation stage through its target gene, that is forming LIF, that is leukemia inhibitory factor. Okay, it is leukemia inhibitory factor is a multifunctional cytokine, so it can cause any activity. So this LIF actually plays a crucial role in the blastocyst implantation, that is implantation of the blastocyst. Okay, so this P53 is regulating LIF in the implantation stage. So this is studied in the mouse model. Possibly it is helpful for humans also. And it is also suggested that recombinant human LIF, it will improve the implantation rate in the women with the unexplained infertility. So women with who are having infertility because of the unexplained cause, we can try this LIF, recombinant LIF, which is derived from human beings. Now coming to the third factor, that is insulin-like growth factor 1, that is IGF-1. It is also called somatomedin C. Somatomedin C. It is a hormone similar to molecular structure insulin. So this molecular structure and insulin molecular structure are almost the same. And this IGF-1 plays important role in childhood growth and has anabolic effects in the adults. In 1950, it was called as sulfation factor because it stimulated the sulfation of cartilage in vitro. And in 1970s, it is called that NSILA, that is non-suppressible insulin-like activity. We are not going into detail, but it is so much to learn about this somatomedins. Next, coming to the TGF, fourth one, that is transforming growth factor. This is having two subtypes, TGF alpha and TGF beta. TGF alpha is upregulated in some human cancers. It is produced in macrophages, brain cells, keratinocytes, and induces epithelial development. It belongs to the EGF family. So it belongs to the EGF family and causes human cancers. It induces epithelial development. Now coming to the TGF beta. It exists in three subtypes, that is TGF beta 1, beta 2 and beta 3. These are upregulated in a Marfan syndrome and some human cancers also, and plays crucial role in tissue regeneration, cell differentiation, embryonic development and regulation of the immune system. The isoforms of the transforming growth factor beta, that is this one, these three. Actually, this one, TGF beta 1, it is thought to be involved in the pathogenesis of preeclampsia. They will act through TGF beta receptors. They are single pass serine threonine kinase receptor. These TGF beta receptors are single pass receptors. That is, they will pass the cell membrane only one time. Now coming to the fifth one, fibroblast growth factor, FGF. We have 23 types of FGFs. Members from 1 to 10 will bind to fibroblast receptor. Members from 11 to 14, they will bind to intracellular fibroblast receptor. And some important points only we have seen. Human FGF18 is involved in the cell development and morphogenesis in various tissues, including the cartilage development. FGF is critical during normal development of both vertebrates and invertebrates, and any irregularities in their function leads to developmental defects. And they are most commonly mitogens, but they are having regulatory, morphological, and endocrine effects also. They have been alternately referred to as pluripotent growth factors. So we can call them as pluripotent growth factors and promiscuous growth factors. Why we are calling like this? Because they have multiple actions on multiple cell types. So these are having many actions on many cell types. So what is this promiscuous actually? This is a term regarding biochemistry and pharmacology concept. That is how a variety of molecules can bind and elicit a response from a single receptor. So receptor is single. Molecule, these molecules will come and to attach to those receptors and how they are eliciting different kinds of responses. So that is called promiscuous effect. It will act through four types of receptors, that is FGF receptor 1, 2, 3 and 4. Now coming to the last one, sixth one, PDGF, platelet derived growth factor. It regulates the cell growth and division. In particular, PDGF plays a significant role in blood vessel formation, the growth of blood vessels from already existing blood vessel tissue. So it is helpful for the formation of blood vessels. And it is also a mitogen, that is proliferation. It will cause proliferation of the mesenchymal cells, especially the fibroblasts, osteoblasts, tenocytes, and vascular smooth muscles and mesenchymal stem cells. Okay, and it is involved in chemotaxis. That is, what is the meaning of chemotaxis? That is directed migration to that particular area. So, what will migrate? Mesenchymal cells. The directed migration of mesenchymal cells. Okay, because of the PDGF. And PDGF will act through a act like a dimeric glycoprotein, and it is also having two subunits, PDGF AB, and they may dimerize like AA, AB or BB. It is a heterodimer, it is a homodimer. The receptor for PDGF is PDGF receptor. Okay, it is classified as a receptor tyrosine kinase. It is a type of cell surface receptor. So it will act through PDGFR receptor. So we have discussed factors that are promoting neuronal growth. So various factors will affect the neuronal development, growth and survival. What are those? Those are classified into broadly into two groups. They are neurotrophins and other factors which are affecting the neuronal growth. So first we have discussed the neurotrophins. So these are actually proteins. They will provide trophic support to the neurons. What is meant by trophic support? They will promote the nerve growth and survival. And also they play a role in the neuronal plasticity. So in that first we have discussed the nerve growth factor. Then we have discussed the BDNF, that is brain derived neurotrophic factor. Then we came to neurotrophin 3. Then we came to neurotrophin 4 and 5. Then we have seen the type of receptors on which these neurotrophins will act. That is TRK ABC receptors. Through three major mechanisms they will act. And these neurotrophins will have high affinity for TRKA receptor. Then they will act through P75 NTR receptor also, but with a low affinity. And this P75 is also causing three types of signaling mechanisms. So those signaling mechanisms also we have seen. Then we came to other growth promoting factors, okay, other than neurotrophins. So they are ciliary neurotrophic factor, leukemia inhibitory factor, insulin-like growth factor 1, transforming growth factor, fibroblast growth factor, and platelet-derived growth factor. So through which receptor it is acting and what it is causing we have seen. The next video is about regeneration and regeneration of nerve fibers.